Hi everyone, welcome to our last but certainly not least session of the UFAW conference. My name is Jian Chu. I'm currently a postdoctoral fellow at Chagas and University of Veterinary Medicine, Vienna. It gives me great pleasure to introduce our first speaker, who is the joint winner of this year's UFAW Early Career Researcher of the Year, Dr. Sara Hintze, who will give a talk on boredom in pigs. So looking forward to her talk.
Hello everyone and welcome to my talk on pig boredom. To avoid any disappointment, I would already now like to say that in this talk I will focus. Hello everyone and welcome to my talk on pig boredom. To avoid any disappointment, I would already now like to say that in this talk I will focus on our conceptual approach of how to study boredom-like states in pigs. I will not present any results yet, and I thought this is important for you to know from the beginning on. So what's it all about? Erich Fromm, a German-American psychoanalyst, philosopher, and social psychologist, once said that, "I'm convinced that boredom is one of the greatest tortures. If I were to imagine hell, it would be the place where you were continually bored." What needs to be emphasized here is that Fromm refers to chronic boredom, not the boredom you may, if you're lucky, experience on a Sunday afternoon, but chronic boredom that probably no one of us really knows, and that is linked to depression, alcoholism, gambling, and suicide. Fromm also stated that man is the only animal that can be bored, and the question, of course, is, was he right? He might have been right if we as humans had not started to impact on the lives of animals in the way we have done in the last decades. Of course, we can find such conditions as we see them here on the pictures for many farm species, but for today, we will focus on pigs only. Already during my master's thesis, I was very curious to find out how to identify indicators of boredom-like states in pigs and what they mean for pig welfare. And I was very happy when much later I received a grant from the Austrian Science Fund to start exploring this topic. Of course, if we want to study boredom, we need to know what boredom actually is. And unfortunately, there is no agreed definition of the construct or well-developed instrument for measuring it. There is no comprehensive theory of its causes, and there's uncertainty about its consequences and importance. And this definition does not refer to non-human animals, but to humans. So where to start if we want to study boredom in pigs? There were two main challenges we were faced with during our experiment, as always when aiming to study affective states in non-human animals. The first challenge is to make sure the pigs we study are actually bored and that we have a good non-bored control group. And then, secondly, we need to find ways how to identify indicators of the affective state of interest, in our case, chronic boredom, and what it means for pig welfare. So let's start with the first challenge, which refers to the experimental design. Given the conditions of the farm we worked at, we aimed to create as contrasting situations as somehow possible. We had two batches with a total of 8 enriched and varying pigs, pens, as you can see on the left, and 8 barren and monotonous pens, as you can see on the right. In the enriched pens, we had different rooting materials, toys that were changed every day, and other enrichment material, for example, Christmas trees in our last batch, that was given twice daily to keep these pigs busy and entertained. Half of the pigs stayed in these conditions for the whole duration of the experiment, which lasted from weaning until slaughter, so around 5 months. The other half of the pigs changed the conditions in the middle. So pigs moved from enriched housing to barren and vice versa. This switch in housing conditions was aimed to create an even stronger contrast by switching from enriched to barren, a switch known to be quite drastic for pigs, and to check if potential boredom indicators are reversible when pigs move from barren to enriched housing. Moreover, from human psychology, we know that it is not only the conditions that determine if an individual becomes chronically bored, but also their personality. Thus, we aimed to identify the pig's coping styles by testing them in a back test, where we measured, when we put the pig on its back, as we can see here on the picture, where we measured latency to the first struggle, the frequency of struggles, and the duration of struggling. And we hypothesized, again, based on literature from humans, that pigs with a more proactive coping style should show stronger signs of boredom compared to pigs with a more reactive coping style. Piglets were tested in the back test while still suckling. They were tested twice during the suckling period, and they were then allocated to the treatments in a balanced way. So let's come to the second challenge, the one about identifying indicators of boredom and finding out what they may mean for pig welfare. Luckily, as we all know, affective states do not only consist of the feeling or the subjective experience, but they also go along with changes in behavior, physiology, and cognition. And these changes we can detect, and we can try to improve the feeling, the subjective experience from them. And this feeling could potentially be, for example, chronic boredom. In our experiment, we focused on the behavioral and cognitive component. But for time reasons, I will only focus on the cognitive component today. If you have any questions about the behavioral parameters, please ask me later or please get in contact after I have finished this talk. So what did these cognitive approaches look like? In order to be able to study boredom-like states, we of course need a definition of what boredom actually is. And as we have seen before, it is not so trivial. However, we are lucky because in animal welfare science, Becky Maher and Charlie Byrne came up with definitions that can also be operationalized, and we tried to capture most of these different aspects of the definitions in our experiment. So let's go through them step by step. Quite obviously, boredom is a negative state. It is aversive. To capture the negative valence, we ran a self-initiated spatial judgment bias task on a subset of our pigs. Unfortunately, due to the time, I cannot go more in detail here, but I think most people will be familiar with the principal design of the judgment bias task. And of course, please feel free to ask again later on. Boredom has also been characterized by an increased generalized interest in diverse stimuli, so stimuli of different valence. And here, our approach was based on studies by Becky Maher and Georgia Mason, and later others were involved here as well, who predicted, again, based on knowledge from human psychology, that boredom would be characterized by a high interest in all types of stimuli, so even ambiguous and even negative ones. And this would be a way to differentiate between boredom-like states on the one hand, and apathy and depression on the other hand. Apathy should be characterized by a reduced interest in all kinds of stimuli. So you're not interested even in the positive, the negative, the ambiguous one. And depression, with anhedonia as a key symptom, there should be a reduced interest in positive stimuli. So you should be able to differentiate between these different negative states based on the interest animals show to stimuli of different valence. First of all, we needed to validate the valence of different stimuli. So which ones are actually positive, neutral, ambiguous, and negative for pigs. And we did this with an approach avoidance paradigm. And you can see here some pictures of the apparatus we used to validate these stimuli. This apparatus consisted of a starting box, a long runway, and what we called an avoidance room where pigs could go if they really did not want to be exposed to a stimulus. And based on all the stimuli we tested, we chose the most distinct ones for the boredom project, and hope with looking at the behavior, the responses of the pigs towards these stimuli, that we can then take this to differentiate between boredom, apathy, and depression. Looking at Charlie Byrne's definition for animal boredom, The first one is that the individual should be motivated to experience anything different than currently possible. Here we used what we call the so-called engagement task, which is based on a study by Wilson et al., which was published 2014 in Science, in which some of the participants of their study, who were college students, preferred to administer themselves electric shocks rather than doing nothing during a 15-minute, only 15-minute waiting period. So they gave themselves electric shocks, even though they said before that they would pay money not to get these shocks. So they were immersive. Based on this finding, we asked whether pigs would also work that means pressing a buzzer for a neutral or even negative consequence when unengaged in a deprived environment. No worries, we didn't use any electric shocks, but we rather used a strong air puff as a negative stimulus, and the pigs could always choose freely if they wanted to work for the different outcomes, which was a positive, a neutral, and a negative one, or if they didn't want to work for it. Last but not least, we were also curious to find out if time drags when pigs are bored. As we know it from ourselves, when we are bored, time goes by much more slowly. And to this end, Christina Kuhl, our PhD student, developed a very nice task, a so-called temporal bisection task. In this task, pigs are trained to differentiate between a short tone, in our case 1 second long tone, and a long tone, in our case an 8 second long tone. It's self-initiated trial, so what you will see in the video in a second is that the pig initiates a trial by touching the green bottle you can see here, and then needs to make a decision once the tone is gone. And I will play now the video showing you the pig. Here you can see that the pig chose correctly the right door after the 8 second tone. And here it went correctly to the left door after the short tone of 1 second. Once the pigs have learned the task, this takes quite a while, they are confronted with a tone of intermediate duration, and then they need to make a decision if they want to go to the side associated with the short tone, in this case the left side, which would be indicative of the time running by fast, like when we have fun, or the long tone, in this case on the right side, which would indicate time dragging, like when we humans are bored. So bringing it all together, we hope to study the effect of both housing treatment and coping style on the behavioral and cognitive component of boredom. But for me, something is important as well. I really hope that we will be able to characterize the individual pigs more in detail, because then this is what is very nice, is that we have really a lot of information per pig. And I really hope we can look at a bigger picture, not only on the effects of housing and of coping style, but also to look for associations between the different results we get to be able to characterize the individual pig more. Of course, all this work would not have been possible without the hard work of the whole Boredom Project team, which I would like to thank very, very much. And of course, it would not have been possible without our wonderful and brilliant protagonists. And this is something I also want to mention because it is rarely mentioned, but our pigs became more than just our study subjects, which I say here in brackets, unfortunately. As you can see here, the interactions or the relationship between us and the pigs was quite intense. And this is after data collection, no worries. We usually did not interact with them like that. But it made it hard to let them go after working for 5 months with them. And with this, I would like to thank you very much for your attention and, of course, the funding bodies for supporting this study. And I'm very curious to hear the questions you have. Thank you very much.
Thank you, Sarah. That's a great talk. Can you now turn on your video so that we can all see you? And can you hear us loud and clear?
Yes, thank you.
Okay, so I will, that's fantastic. So I will start with the first question. So there's a question about personality and boredom. And could it be that the expression of boredom differs between the animals rather than the experience? That is, reactive and passive pigs may express boredom in different ways.
Yeah, that is a very good point, and this is something super understudied. I mean, everything in this field is very much understudied, and that's why we tried just the first step to, I mean, it's also not personality. I am aware in the last slide I said personality, but it's of course only the coping style, so it's not reflecting the whole personality. It's also not reflecting individuality. It was just the first step, trying to think about which of the tasks we have at the moment available and validated, more or less, are connected best with what we know from humans and about human boredom. And that's why we decided to go for the back test and the proactive and reactive coping style. But there's definitely much more to it. Yeah, I completely agree.
Can I just follow up very quickly? So what other personality traits do you think that may actually associate with their expression of boredom?
Whether then boredom is expressed differently?
Yes, like whether the more bold or other personality traits. Just
it's very difficult to predict at the moment, to be honest. I mean, why we came up with these hypotheses is that we know that proactive animals tend more to have this routine formation, are less flexible in their behaviors, and that's how people are also described that are more prone to experience boredom. And of course, it's not only the experience of boredom, but also perhaps how you express boredom, but this is to be looked at. So I completely went over the behavioral part, but for example, we also look at the inactivity patterns of these pigs, trying to see if we can see something in their behavior that is differing between not only the housing treatments, but also the coping styles.
Thank you. So another question also related to individual differences. So were there individual differences in valence attributed to stimuli? And is it possible that this changed with exposure? For example, did some pigs perceive an air puff more negatively than others, or did this change with repeated exposure?
Yeah, also a good point. The problem here is that it's really at the moment we finished data collection, and now it's really about the analysis of the data. So it's hard to say. It's all anecdotal, only what I can say at the moment. But of course, as always, I mean, these pigs are complete as all pigs. They are individuals and they are behaving so differently, and also what they find aversive and what they find more positive differs quite a lot. That's why we did these validation studies before, and also for the in the engagement task where we used the air perf, we did of course some preference studies and so on. But of course you always have differences between animals, which is wonderful because they are individual creatures, but it's also difficult to handle if you want if you want to use the same stimuli for all of them. Whether it changed over time, that is a question. This is one of the questions we need to figure out, because they had different treatment. They were there for different amount of times in these deprived environments. And we hypothesized that as the longer the time you are in this treatment, the more likely you are also going to check out even the negative ones later, like corresponding to what has happened in humans. But as I said, sorry, no results yet.
Jumping for another type of question. So it is very interesting talk. And how feasible is it for assessment of boredom to be integrated into welfare outcome assessment programs, which rely on use of practical measures, for example, the farming situation?
Yeah, good point. I mean, this is definitely a fundamental research project. So that is nothing to be applied yet. We also had quite a lot of TV teams there, and they were always asking, like, what are the recommendations for farmers now? And I was like, no, I'm sorry, that's not for setting up recommendations for farmers. Also, our enrichment treatment was not feasible at all on a larger scale, not at all. It was to have the biggest contrast we could possibly, under these conditions that we had. We didn't have outdoor run or anything that we could create, but it's not about feasibility yet. It's more about understanding if there is something like boredom. It has been mentioned at this conference quite a lot of times already, but what do we know actually about boredom-like states in animals, and what does it mean for these animals, and is it really a welfare problem? I think so, but we are really in the beginning of finding out about this and what this means then in terms of practical application and perhaps changes in legislation. That is, of course, a long way to still go.
Thank you so much, Sara, and then congratulations again. I think we have to move on for now, but please continue the discussion, and if your question is not asked, please put it in Slack so that Sara can continue to answer them. So now we will need to move on to our next talk. Thanks again. So our next speaker is Karolãna, whose topic is promotion of play behavior as a means to improve pig welfare.
Hi, I would like to welcome you to my presentation about promotion of play behavior as a means to improve pig welfare. This project is a part of my PhD thesis, which is on play behavior in pigs. To start broadly about how to improve animal welfare, the traditional approach has a strong focus on reducing negative aspects of animal welfare, such as pain, disease, and distress. However, now it's being acknowledged that while the negative welfare states must continue to be minimized, there should be an emphasis on promoting positive experience. It should be attempted to achieve positive welfare balance, defined as the balance between providing the opportunities for positive experience beyond the alleviation of the negative states. This view at the same time addresses the public concern about the quality of life of farm animals. To get a complete picture, first we need to define what is a positive experience for pigs. Most likely, a positive emotion or affect is generated when an animal has opportunities to respond to motivations, to engage in rewarding behaviors. For pigs, it can be behaviors like foraging, maternal care, human-animal interaction, or play. Now let's talk about play and its relation to animal welfare. It has been suggested that play may reflect the current welfare state of an animal. This has been proposed because play is initiated when the animal is in a relaxed field, meaning that there are no immediate threats to the animal's fitness, and decreases when conditions become more challenging. Play also occurs spontaneously and voluntarily. Furthermore, play has been also proposed to be an indicator of animal welfare because it's easy to recognize with play markers and can be measured non-invasively. To continue about play and its significance for animal welfare, it's been also noted that play may contribute to the promotion of positive welfare, meaning that playing could also cause welfare improvements, not just reflect them. Commonly, play has been linked to the experience of positive emotions in non-human animals, as it has been long in humans. Play is believed to be rewarding and indicate the presence of opioid-mediated pleasurable emotional experiences. It was found that the subcortical brain areas and opioid neurotransmitters involved in the reward system overlap with the brain areas that are engaged in social play in rats. Play can be also a motivating reinforcer. In a preference study with chimpanzees, play was valued as highly as the most favorable food. Play also play a role in the social bonding and strengthening the bonds in groups. Lastly, it's argued that play has a rebound effect and may act as a stress coping mechanism resulting from challenging conditions when animals have new opportunity to play. For example, piglets from barren pens had increased play frequency than piglets from enriched pens when given opportunity to play. This diagram represents emotional experience in two-dimensional space. Every emotion is either positively or negatively balanced and has high or low arousal. Positive affective states are in quadrants 1 and 2, and negative states are in quadrants 3 and 4. Here I want to show that play belongs to the quadrant 1 and is coupled with high arousal and is positively balanced. The position of play is interesting to compare to exploration that is also positively balanced, but its arousal is low. Play behavior in pigs commonly occurs between 2 to 6 weeks and declines as the pig gets older. Hypotheses about the functionality of play say that play aids the development of social, cognitive, and motor skills early in life. Play in older pigs hasn't been reported very often, and its function is debatable. Some say that play occurs during the instability when animal needs to evacuate stress, which would be the rebound effect of play mentioned earlier. However, it can be also argued that if play is intrinsically rewarding, the animal in stress makes itself feel better by playing, which is overly positive. To give you a rationale for the study, researching play as a means to improve pig welfare, we first needed to determine the possibility of promotion of play in pigs beyond the natural window of the development. And because commercial settings offer little stimulation for pigs due to spatial restriction and barren conditions for hygiene purposes, when pigs are given the opportunities to respond to motivational behaviors leading to positive emotions, the negative welfare states, such as boredom and frustration, may be replaced for a joyful activity such as play. Importantly, these findings would link to development of intensive animal-friendly systems where opportunities of play could be used to improve the joyfulness in pigs' lives. The objective of this study was to investigate whether play can be repeatedly promoted and sustained in growing pigs in commercial facilities. It was explored from 10 to 21 weeks of age in Prairie Swine Center, which is a production as well as a research farm affiliated with the University of Saskatchewan. We used 288 pigs and assigned them to 3 pen-level treatments. The pigs were housed in a standard semi-slotted pen that we call the home pen. Control treatment was only kept in the home pen with no other interventions. And then we had 2 play treatments, novelty and playpen. Play was promoted during a play session when novelty treatment was given novel objects to the home pen, whereas play treatment had access to extra space in a larger pen with the same novel objects. Play was promoted 3 times a week, 2 times a day in a 30-minute play sessions. Novel objects consisted of 6 items such as cardboard, burlap, rope, untreated wood, popcorn, and straw. These objects had characteristics favored by pigs. They were manipulable, investigable, chewable, and edible. Their novelty was supported by weekly rotation and once a week with olfactory stimulation with one of the 3 essential oils, garlic, lavender, and thyme. Extra space was offered to play treatment as it was shown to stimulate play in piglets. Play behavior was recorded during a play session on 144 focal pigs. We recorded the play behavior in 3 stages at 11, 16, and 21 weeks of age. We recorded locomotor, social, and object play. Within locomotor play, we had different behavior elements such as pivoting, gambling, head tossing, flopping, or rolling. Social play was a reciprocal play between 2 or more piglets. And object play included object manipulation and shaking and carrying of the objects. However, for the purpose of this presentation, the results will be presented as a total play behavior, which are all the behaviors combined. For statistics, we had frequency and duration of play behavior, and the data were not normally distributed. Thus, we analyzed them in a multi-level regression with negative binomial distribution. The pig was used as a repeated measure, and the predicted means accounted for multiple comparisons with Bonferroni correction. This graph shows the frequency of total play in a 30-minute play session. The y-axis shows the play frequency per pig in 30 minutes, and the x-axis shows the treatment and their ages. There was a significant interactive effect between the treatments and the weeks. Let's first focus on the significant differences between the treatments within the week that are denoted by a star. Compared to control, pigs from novelty and playpen played consistently more frequently in all weeks. Pigs from playpen treatment played more frequently than novelty, but this difference was only significant at 21 weeks of age. Now, if we are looking at the significant differences within the treatment across the weeks, denoted with different superscript letters, we see that the play frequency in all treatments significantly decreased, but novelty and playpen sustained the same level of play at 16 and 21 weeks of age, whereas play in control continued to decline. This graph shows the duration of total play in a 30-minute play session. The y-axis shows play duration in minutes per pig in 30 minutes, and the x-axis shows the treatment and their ages. Again, there was a significant interactive effect between the treatment and the week. Focusing on the significant differences between the treatments within the week, compared to control, novelty played for a significantly greater duration in all observed weeks. Compared to control, playpen played for a significantly greater duration in weeks 11 and 21, and numerically in week 16. Novelty played for a longer duration than playpen at the age of 11 and 16 weeks, but statistically this didn't differ. Regarding the differences within the treatment across the weeks, novelty and playpen sustained the same duration of play across all weeks. Here, I would like to show you an example of behaviors during a play session, first in control, then playpen, and then in novelty. We were able to increase play and sustain the play response in play treatments compared to control. Play decreased over time, but despite this decrease, play was still greater in play treatments. We showed that the intermittent provision of favorable enrichment with novel aspects effectively stimulated play either in the home pen or with addition to extra space in the playpen. Despite the two environments, in the results we saw the same level of play was generated in both play treatments, suggesting that play can be stimulated in the home pen in standard production settings without provision of extra space. The repeated voluntary engagement in play until the late stage of finishing period suggests that the behavior was rewarding and positive for the pigs. Also, as seen in the graphs, a little bit of play occurred in control pigs, which could be to some extent due to animal contagion, because the treatment shared one room. All in all, this study is a proof of concept, and this approach could be refined for practical applications on commercial farms. To conclude, play can be successfully, repeatedly promoted and sustained in grow-finish pigs in standard production settings. Play opportunities could support positive welfare in commercial scenario. The findings from this study could play a role in the development of intensive animal-friendly husbandry systems, where opportunities to play could contribute to positive welfare balance. Importantly, because play is more readily performed in alternative systems compared to intensive systems, this approach could be one way to help improve pig welfare in existing commercial systems. I would like to thank you to our funders, that includes the range of industry and government partners, for the support of this research. Lastly, I would like to acknowledge the staff members in the Prairie Swine Center, as well as the swine welfare team at the Western College of Veterinary Medicine, for the encouragement, help, and support. Thank you for your attention, and with this, I will take your questions.
Thank you, Karolãna. Can you turn on video? Can you hear us?
Yes,
Great.
I can.
Thank you for a very nice talk. I will start with the first question. It is known that stress resistance in pigs is associated with productive features of this animal. Do you consider to study some association between pigs' play behavior and some physiology characteristic of the pig, such as overall health, decrease in number of disease cases, better reproductive quality, etc.?
Thank you for the question. Yeah, so actually in this presentation, I focused on behavior, but we collected additionally different measures. We collected average daily gain to measure some physiological effects of play. And as I said, the PhD project is comprised of different experiments. And actually, my next experiment focuses on disease resilience, so how play can affect disease resilience, and can it improve disease resilience. So I collected the data, and now I am about to analyze the data. So this will come, and I will present the results later.
Great, looking forward to your upcoming results. So another question asks, does enrichment and space affect the type of play? For example, the locomotor, social, and object play observed by you differently?
Yes, so I have analyzed locomotor, object, and social play, and we can see differences in play in the two-play treatment. So as it's expected, more locomotor play was performed in playpen treatment, and more object play was performed in the novelty treatment. So the different environments generate different play response, which will be further investigated. What are the effects? Or yeah.
Thank you. And another question is, might there be some minimum home pen standards below which it might not be possible to stimulate and sustain play? For example, a minimum space allowance per pig or non-slippery floors, etc.
Yes, that's interesting. I would say this experiment was, we deliberately chose a smaller home pen for the practical reason, so we could have 8 pigs in it. But for standard production, it's common practice to have a bigger pens with more pigs. And it can be argued that it's actually easier to stimulate play in a bigger pen with more pigs because they have more space, but definitely it depends on the density of the pen. So if the pen is very crowded, there can be also some agonistic behavior going on, and the spatial restriction can not support the play. So yeah.
And in one of your results, when you were talking about the age effect, it seems quite interesting that in the playpen, there seems to be kind of a decrease at the age of 16 and then later increase again at 21st. Do you have any explanation for that based on your observation?
Yes, I actually do. I was also surprised by this decrease. And I think what happened is because the playpen was outside of the room, and the farm is a functional farm, so there is stuff going through, and so the playpen treatment was more prone to disturbance outside of, like, we couldn't control that. So something might have happened that the pigs were probably scared for the period of time of the recording. Or yes, there was something going on that we couldn't predict and control. So that's probably why it decreased.
Thank you very much. Thanks again for the talk. And then please continue the discussion in Slack. So we're now moving.
Thanks, Jen. Give me 2 seconds while I share my screen. So thank you, everyone. It's been an absolutely amazing couple of days. I'm going to have to go back and watch the videos to truly absorb everything that we've talked about over the last couple of days, because there's been just such an enormous range of really fascinating science, which is, I think, what most people, including me, love about UFAW meetings, that it's really eclectic and you get to learn about lots of science that's way outside your field, but that might be really useful for the work that you do. So thank you to all the presenters. It's amazing to see such expertise and enthusiasm on so many different subjects. And I really appreciate all the work you've put into those fabulous presentations. Just a reminder that the Slack channel is still open to discuss the posters or to post questions about talks that you haven't managed to get answered during the live session. So do head over there and have a look at that Slack channel. And also just a plug for our friends at the Animal Welfare Slack channel, where if you want to discuss more general animal welfare or see some other talks, that's a great place to do that. And there's a link in our Slack channel to that one as well. For those of you who are going to ask us whether you're going to get a certificate of attendance, there'll be an email from GoToWebinar in the next day or two, which will give you a link to download your certificate of attendance. So don't worry if you need one of those, it will be on the way. Just to remind you, we will be holding a conference next summer around about this time, hopefully, which will be a hybrid meeting. It will be online and in person for those of you who want to get together. Hopefully somewhere in Europe. We'll be finalizing the venue as soon as we can, and we'll let everyone know as soon as we know where the conference is going to be next year. But I really hope to see you all either in person or online next year. While we're talking about meetings, although I said how wonderful it is to have really general animal welfare meetings, UFAW also supports some very specific animal welfare meetings. For over 30 years, we've collaborated with the RSPCA to run a meeting about the welfare of rodents used in research. The meeting this year will be on the 31st of October in London. We'll be opening that up for registration in the next couple of weeks, hopefully. So do look out for that one if that's your particular area of interest. And our sister charity, the Humane Slaughter Association, is running a meeting on animal welfare at slaughter and killing also in October this year. The registration is already open for that one at the link down there, but just have a look at the HSA website if you need more information on that one. So that really brings me to the absolute end of this meeting, where it's my pleasure to thank everyone who's made this meeting a success. As I said, all the speakers and poster presenters have put a huge amount of work in. Thank you very much. You're the ones who give us the content that makes these meetings what they are. Thank you to all the volunteer session chairs. It's not an easy job to chair these online sessions and field all the questions and pay attention to the talks. So thank you to those. Thank you to all the folk at UFAW who make this happen. Sam and Liz have been involved in all sorts of organizational stuff. Louisa Dormer is out there tweeting from the UFAW account, frantically trying to summarize all the talks and doing an amazing job. And of course, huge thanks to Steve Wickens, who's been doing all the technology in the background and organizing all the speakers. Steve, if you're there, stick your camera on and take a bow, because you've done an amazing job. So big thanks to Steve, who some of you may not have seen, but he's the one who's made sure that all this has run completely seamlessly for 2 days. So amazing job. Thanks, Steve. And that brings us to one final thing, which is to ask you to sign up to our mailing list so that we can keep in touch with you about future meetings. And the very final slide that I'm going to leave up is just a survey to ask you how you thought this meeting went and whether there's anything we could do better. So if you click that QR code or follow that very short link, which we'll also send out to you or put in the Slack channel, we'd be really grateful if you could tell us how you found the meeting and whether there's anything we could do better in future. So on that note, thank you all very much. Have an excellent rest of your day, whatever time of day it is for you, wherever you are in the world, and we'll see you next year. Thank you very much, everyone.